Today, we're diving deep into what I like to call your body's inner ecosystem, the gut microbiome. For too long, we've viewed our bodies as purely human, but the truth is, we're a fascinating blend of human cells and trillions of microscopic organisms living within us, primarily in our gut. Think of it as your inner garden, that, with the right care, can flourish and contribute profoundly to your overall well-being. This approach aligns perfectly with naturopathic philosophy, understanding the why behind our health challenges by looking at the intricate systems at play. Let's explore this, what some scientists refer to vital symbiotic organ, and learn how to cultivate it for our optimal brain, gut, and immune health. First up, what exactly is this gut microbiome we're talking about? Simply put, it's a vast and dynamic community of microorganisms, including bacteria, fungi, viruses, archaea, and even some parasites that reside primarily in your human gastrointestinal tract. Some researchers even refer to it as a sixth vital organ. While the gut microbiome is not an organ in the classical anatomical sense, it is widely recognized as a crucial organ-like system integral to human physiology and health. Now, when we talk numbers, prepare to be stunned. Your gut microbiome contains an estimated 100 trillion living microbes. This staggering number means that these microbes actually outnumber the human cells in your body, potentially by 3 to 10 times. If we were to scoop them all out and put them on a scale, we'd be looking at about 1 to 2 kilograms in weight, making this microbial organ similar in size and weight to your liver. What's even more fascinating is that it likely exceeds the liver in the number of biochemical reactions it participates in, thanks to its vast genetic potential. And it's not just about the sheer volume, it's about the incredible diversity. To date, over 1,000 different species have been isolated from the human gut. Although a typical Westerner usually harbours around 160 different species, each one of us has a unique microbial fingerprint, shaped by our genetics, early life exposures, diet, lifestyle, and environment. Most of these microbes live in a mutually beneficial symbiotic relationship with us, meaning both we, the human host, and the microbiota benefit. This is why many researchers now consider humans super-organisms, or holobionts, a complete unit of biological organisation composed of a host and its microbiota. So what exactly do these tiny residents do for us? The functions of a healthy microbiome are absolutely crucial for maintaining our health, and they span far beyond the functions of the gastrointestinal tract. Let's start with digestion and nutrient synthesis. Our human enzymes can't break down all the complex carbohydrates from our diet, especially dietary fibers. That's where our gut microbes step in. They ferment these indigestible carbohydrates, producing incredibly beneficial compounds called short-chain fatty acids, like acetate, propionate, and most notably, butyrate. Butyrate is a powerhouse. It's the primary energy source for our colon cells, essential for maintaining the integrity of our gut barrier. It has anti-inflammatory effects, improves insulin sensitivity, and even influences the blood-brain barrier. Beyond energy, our gut microbes also synthesize essential vitamins, including many B vitamins like thiamine, riboflavin, niacin, biotin, and folate, as well as vitamin K2, which we absorb in the colon. They even play a role in enhancing the solubility and absorption of minerals like calcium and magnesium. Next, the microbiome is fundamental for immune system development and regulation. From the moment of birth, our microbiome acts as a trainer for our immune system, teaching it to differentiate between beneficial microbes, friends, and harmful pathogens, foe. This early education is absolutely critical for developing a balanced and effective immune response throughout our lives. The gut microbiota profoundly impacts our inflammatory milieu, either contributing to or actively countering systemic inflammation. It influences mucosal IgA production, the development of our thymus and spleen, lymphocyte function, and natural killer cell activity. And finally, our microbiome is essential for maintaining the gut barrier. This protective barrier along our gut lining is our first line of defense, a highly selective interface between our internal environment and the external world. Beneficial bacteria physically form part of this barrier and also prevent the overgrowth of harmful pathogens by competing for nutrients and attachment sites on the intestinal wall. When this barrier is often compromised, we call this leaky gut. It allows pathogens and toxins to enter the bloodstream, potentially triggering systemic immune responses and inflammation throughout the body. Now what happens when this delicate inner garden, this complex ecosystem, goes awry? This state is known as dysbiosis. Dysbiosis can manifest as a loss of beneficial bacteria, a reduction in overall diversity, or an overgrowth of potentially harmful bacteria. And the consequences are far-reaching. Dysbiosis has been linked not only to a myriad of gastrointestinal conditions like inflammatory bowel disease, or IBD, irritable bowel syndrome, or IBS, non-alcoholic fatty liver disease, liver cirrhosis, and small intestinal bacterial overgrowth, but also to a growing list of systemic diseases. We're seeing strong associations with conditions affecting the brain and mood, such as Alzheimer's disease, anxiety, depression, and multiple sclerosis. It's also implicated in metabolic disorders like obesity, type 1 and type 2 diabetes, and many autoimmune conditions such as rheumatoid arthritis. The research over the last decade has truly highlighted the microbiome as a key driver in these disease states. So, what are the modern life factors contributing to this imbalance? Unfortunately, many are prevalent in our contemporary society. First one we need to talk about is diet. The typical Western diet, high in saturated fats, refined sugars, and processed foods, and low in fibre, is a primary culprit. This diet can drastically reduce microbial diversity, increase inflammation, promoting gram-negative bacteria, and negatively impacting short-chain fatty acid production. Even common food additives like emulsifiers, for example, carboxymethylcellulose and polysorbate 80, can disrupt the gut's mucus layer, increasing permeability. And those artificial sweeteners, saccharin, sucralose, and aspartame, can alter gut microbiota composition and function, potentially leading to glucose intolerance and reducing beneficial species like Akkermansia, Eosinophilia, and Bifidobacteria. The next factor is medications. Perhaps the most well-known disruptors are antibiotics. They can cause significant and long-lasting damage to the gut ecosystem, reducing diversity and wiping out crucial beneficial bacteria like Bifidobacteria. Even proton pump inhibitors, or PPIs, commonly used for reflux, have been shown to markedly decline microbiota diversity in as little as 7 days. The final contributing factor is lifestyle choices. High stress levels and insufficient sleep are also known to negatively impact the microbiota. A lack of moderate exercise can reduce microbial diversity. Other significant factors include our age, genetics, and even our mode of birth. Given this complex picture, how do we cultivate a healthy inner garden? The answer lies in fostering microbial diversity, which is widely considered a key hallmark of a healthy and resilient gut. High diversity supports robust digestion, efficient nutrient absorption, strong immune regulation, and a greater ability for the microbiota to recover from disturbances. From a naturopathic perspective, our focus is always on holistic integrated strategies. Number 1, dietary foundations. This is paramount. A predominantly plant-based diet rich in fibre, diverse colours, and polyphenols is your best friend. Dietary fibres found in fruits, vegetables, whole grains, and legumes are the primary food source for your beneficial gut microbes, driving the production of those short-chain fatty acids that are so vital. Prebiotics, a specific type of dietary fibre like inulin, fructooligosaccharides or FOS, galactooligosaccharides or GOS, and human milk oligosaccharides, HMOs, selectively enhance the growth of beneficial bacteria such as Bifidobacteria and lactobacilli. Polyphenols abundant in plant-based foods like berries, tea, coffee, and wine also nourish beneficial bacteria like Lactobacillus. And if that sounds unfamiliar like it did to me when researching this podcast, it's because it's a recent name change as it was formerly known as Lactobacillus plantarum, commonly used to treat IBS, especially the 299B strain. Polyphenols also nourish other beneficial bacteria such as Bifidobacterium, Rosviria, Faeculobacterium, and Akkermansia muciniphila, a very important bacteria that maintains mucosal integrity. Many polyphenols are too large to be absorbed directly and rely on microbial metabolism in the colon to become active and absorbable, highlighting the crucial role of your gut microbes in their medicinal action. This is a true win-win scenario for both host and our little microbial friends. Resistant starches, found in things like raw potatoes, green bananas, cooked and cooled rice or potatoes, also feed butyrate-producing bacteria, boosting short-chain fatty acid production. Butyrate can increase the number of beneficial bacteria, reduce inflammation, regulate gut motility, support metabolic health, and has even demonstrated neuroprotective properties. The second strategy is lifestyle adjustments. Number 1, moderate exercise improves microbial diversity. Consistency is the key here, and if possible, outdoors is even better. Which brings me to my next point, outdoor exposure in nature. Research clearly backs this up. A study compared urban environments to natural environments, and nature clearly wins out. So get your forest walking in. 3. Aim for adequate sleep, ideally more than 7 hours per night. However, total sleep time over a week is generally a better way to gauge quantity. Number 4. Implement stress management techniques like meditation, as stress negatively impacts your microbiota. Number 5. Where possible, minimize unnecessary use of medications and dietary additives known to disrupt the microbiome. The next strategy is thoughtful use of herbal medicines. As a clinical naturopath, I often get asked about herbs. This is where an integrated approach is vital. Herbs can nurture and nourish. Many herbs contain polyphenols and polysaccharides that act as prebiotics, feeding beneficial bacteria and enhancing short-chain fatty acid production. For example, a herb such as Cotinopsis has polysaccharides that can increase beneficial bacteria like Bifidobacterium and Lactobacillus and Akkermansia, while decreasing harmful ones, and it can also improve the bioavailability of other active compounds within the herb itself. This also highlights why the form of the herb matters. An ethanol-based tincture may contain different constituents than a water-based decoction or powder, and thus have a different impact on your microbiome. In the previous podcast, I go into more detail about herbal medicines and how they impact the microbiome. I also discuss the impacts of antimicrobials, some beneficial with great selectivity, keeping your microbiome intact, and more importantly, I tell you what you need to avoid to prevent wiping out complete populations of beneficial bacteria. So go check out that episode for those details. Finally, I want to discuss probiotics. While valuable, current generation probiotics have limited capacity to permanently colonize the gut or fully restore an imbalanced microbiota on their own. However, specific strains can indeed increase populations of certain beneficial microbes, such as Faecalibacterium plasnitii or various strains of Bifidobacterium. While many medical professionals incorrectly like to point out probiotic supplements have no benefits, there continues to be mounting evidence that shows the complete opposite. Therapeutic strains such as Lactobacillus rhamnosus GG have shown many benefits, such as reduction in IBS symptoms, relief of constipation, coronary artery disease risk reduction, improvement of type 1 diabetes management, and even the prevention of COVID-19 symptoms. Often critics will reference studies that don't look at specific strains. For example, if a meta-analysis was only species-specific and not strain-specific, then all Lactobacillus plantarum species could be pulled in together. Lactobacillus plantarum strain 299V has been shown to effectively reduce IBS symptoms in several clinical trials, whereas administration of Lactobacillus plantarum strain MF1298 was found to actually worsen IBS symptoms. Other Lactobacillus plantarum strains have shown no effect on IBS outcomes. Therefore, in this example, by not being strain-specific, the data may show no benefit and potential harm from using a probiotic in the treatment of IBS, drawing the conclusion you should not use a probiotic to treat IBS, when in fact, by using the correct strain, in this case, Lactobacillus plantarum 299V, you can effectively reduce IBS symptoms. This subject clearly warrants its own podcast, and if you would like me to discuss more on probiotics, please let me know. So as you can hear, your gut microbiome is not just a collection of germs. It's a dynamic, vital, and complex organ. By understanding its functions and the factors that influence it, we gain immense power to shape our health. Cultivating this inner garden through a diverse polyphenol and fiber-rich diet, smart lifestyle choices, and thoughtful use of herbal medicines can profoundly impact your digestion, your immune resilience, and even your mood and cognitive function. I'll put a link to any references in the description. Join me next time as we delve deeper into specific strategies for supporting your gut, brain, and immune health. Thanks for tuning in.